Hi, good afternoon, everyone. Thanks for sticking around to the almost very end. So I'm Laura Koivusalo. I'm the CEO and founder of StemSight. And at StemSight, we are curing blindness with stem cells. So briefly as a summary from our company, so we are a preclinical stage private biotech based in Finland. And we come from a spin-out of Tampere University, where we come from Professor Heli Scottman's lab. And Professor Heli Scottman has been one of the leading ocular stem cell therapy developers across the world, first focusing at back of the eye applications, but now lately more in the corneal cell differentiation. And that's where StemSight is currently focusing and is our sort of unique therapeutic advantage. So our company's technology builds on our expertise in iPSC differentiation and biomaterial carrier design to actually not just inject cells and hope that they'll end up in the right place and integrate, but rather provide ready-made tissue structures that are functional from the very beginning. We come from the university and our IP has been developed there, but we do have full and exclusive ownership of all the IP and patents around our technologies. To date, we've raised about €1 million in equity from local VCs and family offices, and we've more than doubled that with non-dilutive funding. So our scientific founder team comes from the same research group, academic group. We've been complemented by additional experts in iPSC space. One of our stars is, of course, our head of R&D and CMC, Dr. Sarita Panula, who's been traveling around the world with iPSC and pluripotent stem cell therapies, starting from Stanford, going to Kyoto with Yamanaka himself, then back to Sweden in Karolinska, and then at Procella. Now she's been leading our R&D and CMC activities as employee number one. Last fall, we also hired our first commercial person to the team, so Dr. Ross McDonald, who some of you in the room might remember from Synata Therapeutics. So for the past 10 years, he was the CEO of Synata, who was the first company in the world to advance their allogenic iPSC-based therapy to the clinics. Our board of advisors come from pharma industry, stem cells, regulatory affairs, and ophthalmology. At StemSight, our remit is simple. We replace lost cells. So we provide curative one-time treatments with our proprietary regenerative cells. We aim to provide off-the-shelf tissue products using HLA hypoimmunogenically edited iPSCs and our biomaterials. And we do see this as a sort of a platform technology, of course, because iPSCs can have the potential to make any type of tissue, but we are laser-sharp focused currently in corneal blindness. And why corneal blindness, you might ask? It's, of course, one of the in regenerative medicine, of course, we try to engineer different kinds of cells to replace cells in the body, to replace lost function. But in the cornea, there's actually a well-established clinical precedent of actually seeing functional sight restoration by replacing lost cells. So our first lead indication is limbal stem cell deficiency. Limbal stem cell deficiency is a rare condition, mostly affecting young and working age people. So limbal stem cells are located around the edge of our corneas, where they constantly produce new corneal epithelial cells to replace cells that we shed away with every blink of our eyes. But they can be lost, for example, due to burns, chemical injuries, mechanical damage, radiation, Stevens-Johnson syndrome, and some genetic conditions. And this actually results in the cornea no longer being able to heal itself, and thus the surrounding tissue starts to cover the cornea, including blood vessels. And this leads to a progressive vision loss and blurriness of vision. And it's actually also a very painful condition that's not treatable with corneal transplants. Currently, the only available therapy for these patients is autologous limbal stem cell transplantation, if they still have one healthy eye left. So that's where the clinical precedent comes from. And this autologous therapy has been approved in Europe since 2015, by the name of Holoclar. Since we're in Italy, I think it's pretty fitting because this is the home of Holoclar. And there are similar autologous transplantation therapies all across the world being developed. So why go with an iPSC-based approach? Well, of course, autologous therapy can only treat patients who still have a healthy eye left. But what we can do is we can actually treat patients who are blind, who don't have any of these limbal stem cells left. And with limbal stem cell deficiency, even though it's an orphan indication, because the patient group is very young and at an early age, average age of the treated patients is only 34 years, so there is a high pricing potential for these therapies, as we've actually done health economic assessments with Cell and Gene Therapy Catapult to estimate the pricing potential for this therapy. Based on published data, we know that there are about 36,000 eyes to treat in Europe and the US combined, bringing our market size already to $14 billion. So, limbal stem cell deficiency, our lead indication, unmet need. But our second pipeline product that we already have IP on is aimed for Fuchs endothelial dystrophy and other corneal endothelial dystrophies. So that is a condition where the inner lining cells of the cornea have been lost, and most often caused by aging. And that's also a progressive loss of vision. So currently, this is the leading cause of corneal transplantation worldwide. And currently, there are 70 times more patients than there are cornea donors available. So this corneal donor shortage is something that we can address with our iPSC platform. So our products are based on our years of expertise in differentiating different ocular cells and also developing biomaterial carriers to make functional tissues with these cells. And we can manufacture all of our products for all of our patients from one single iPSC line. So these are the two therapies that we currently have in our pipeline, and we are looking to broaden our pipeline in the future. So as an overview of our process, this is how we see the manufacturing of these two different therapies. So starting with one iPSC cell line, we can, as a master cell bank, we can start our differentiation into these different cell types. More now zooming in on the limbal stem cell differentiation, which has been more in our company's focus since we established, and we've worked heavily on that, and I'm gonna give you some highlights on the manufacturing side of things, just in a couple of slides. So there is actually a two-step process in our differentiation. So we actually start the differentiation towards limbal stem cells, but we halt that in a specific progenitor cell type, which we then enrich and expand. It's about 35 days of processing from the thawing of the first iPSC vial into the batch of cells, of limbal stem cells that we can then cryostore. For each patient, we'll then thaw one vial of the cells onto our proprietary carrier material, and then that will be shipped fresh into to the operating hospitals. So as sort of a scientific side note on why we are targeting this particular cell population and why it's important. So within the within the limbal stem cell biology, it's actually, of course, been identified from adult tissues, taking pieces of tissues from living people and also from cadavers. And of course, working with adult cells and fully matured adults, it's also a limitation of our understanding around these kind of progenitor cell types. But when we're using iPS cells, we sort of get a window of opportunity to look at the different stages of differentiation of these cells. And what we actually found out was that when we start the differentiation towards the limbal stem cells, that we were aiming for was at the bottom of the screen, you see the day 24 population, which is this sparsely distributed, very sort of bright PAX6 and P63 alpha positive cells, which are one of the hallmarks that are currently used for adult limbal stem cells. But actually what we found out was that at day 11 in the differentiation process, we find this transient expression, high expression of ABCG2 around the membrane, cell membrane. And this is actually the pool of cells that we actually can now enrich for our therapeutic population. And then we've actually now had the opportunity to look at this expression in fresh cadaver tissue as well. And there we also see this kind of marker expression profiles. When we look at very fresh cells and go really deep into the layers, we do see that these kind of cells do exist, but they are quite sparse. In terms of in vivo proof of concept, so we work together with several academic groups to establish animal models in limbal stem cell deficiency. It's actually a very, somebody noted earlier about the difficulties of using human cells in animals, and that's definitely something that we've seen. So we have short-term animal data under heavy immunosuppression so that our cells wouldn't be rejected in these animal models in the inflammatory eyes. But we've worked with rabbits and mice in the past, and there we do see that compared to untreated eyes, which are wounded across the limbus, when we transplant the cell product, it does maintain the vasculature at bay around the edge of the eye, and it does remain clear enough to see the pupil through the eye. But more importantly, we'll look at histology. So we've seen that the cells already at this day 6, day 7, do start to form the stratified epithelium of the cornea with correct marker expression at the basal level, as we'd want to see in the mature eye. So, but most importantly, what we've done at the company's side is focus on the manufacturing, which I know is a major bottleneck for many of us in the industry. So, our limbal stem cell differentiation process that we inherited from the academics, we transformed that into a commercially feasible process. And we first, of course, worked with different cell lines, different iPSC lines to test the differentiation process. So far, we've worked with 3 different iPSC lines, and our differentiation works very robustly in each of these lines. We use our ABCG expression as our main QC criteria, and then this was with our research grade line where we did process optimization, about 80–90% pure population of cells at the end of our process. But now, when we've moved on to a clinical grade GMP iPSC line with GMP reagents, we actually have increased the purity to 99.9%. And more importantly, the process that we have is also scalable. So this is our current process that we think that will suffice for our phase 1 to 2 clinical trial using our GMP reagents in a semi-closed system. And we can already produce 100 patient doses per batch in this typical 2D culture condition. And then at the last phase, we'll do the product assembly, thawing 1 vial of the cells onto the carriers. But we also envision what scale-up will look like to go up to 350 vials. And there we'd actually maintain the first part of the process constant. So in terms of timeline, so we have been established a couple of years now. We've raised $2 million altogether $2 million in funding. We're now in the process of raising another seed round to get us through the preclinical feasibility state to put together a full preclinical package and do the rest of the CMC to GMP work to really make us ready for our bigger Series A, getting us through the first in man for our lead asset. Thank you, and there is a minute for questions.